So we'll begin. Before I begin, any questions, concerns, or anything that's remaining from the last class? We'll finish what we started last class, but just to give you a very quick glimpse, we started talking about PCR. We started talking about microarrays, and what we're going to do today is we're going to finish talking about how sequencing happens. And this is in some senses as hot as it gets. One of the technologies that I'm going to talk about towards the end, hopefully we will have time, is just a couple of years old. It gives you a $200 genome. So far we are trying to touch into all sorts of modern aspects of where the field is. It's very sort of easy also to get outdated in this field, actually, in some sense. But hopefully we'll not do that. That's why there's no textbook for this class, because it's hard. It's every new day there's a new paper. Every new day there is a new technique that comes out. Very, very hard to capture that as part of the. Okay, I don't know what happened. Just give me 1 second. Any questions from last class? Okay, so I thought it's a good time to actually start with a music. I just want you to listen. It's very cool music. Any idea what is going on there? Very cool music. The only time I've heard this kind of music is in, well, at least it used to be in my era, was this TechFest videos. TechFest would have a special launch of its videos 10 years back, and it would have all sorts of, I don't even know the genre of this music, but some sort of electronic EDM stuff. Very fancy. But what was going on in the video? Any ideas? Okay, but what was happening? Like, what is the process? What are we doing? DNA sequencing, right? So yeah, we are sequencing DNA, right? It's a field of biology, but there is current, there's electrical engineering. How from sequences we go to current, and from that current how we go to sequences is actually an electrical engineering and a computer science problem. We will try to solve it and see where we go along. Okay, we will formally begin. This will, of course, re-feature again as we talk along. So last class we had discussed about Sanger sequencing. Is the concept of Sanger sequencing clear? Because that is forms the basis of your questions. Are cleared? Okay. Any questions about Sanger sequencing? The central premise there is you add certain labeled nucleotides, and you mostly add normal nucleotides. So you're adding your dideoxy ones, which are not normal, because once incorporated, they will not allow your DNA sequence to extend any further. And you're adding 90% of the stuff that you're adding is normal, so it will keep getting incorporated. And as it keeps getting incorporated, at some point, by the way, people who are coming in late, you missed a very interesting music. I really want to play it along again because that music itself is so exciting, but we'll do this at the end of the class. But the idea here is the if you incorporated bases which cannot extend, you can actually do a read through and figure out what sequences you are going to actually sequence. And whatever we discussed today for the next generations of sequencing, this is, by the way, often referred to as the first generation of sequencing, because this is 1970s. This is all we had if we wanted to determine the sequence of a short fragment. We also discussed why it had to phase out at some point, in the sense that it could only do 100,000 bases per day, which is extremely slow. Think about doing this for 3 billion base pairs. You'll spend your lifetime doing this. And it's only accurate for fragments which are less than 700 base pairs. What we're going to discuss today, we will try to talk about it as to how this was extended. Where do we stand now overall? Any idea, any questions about Sanger sequencing? This part is clear, everyone? This forms the premise of what we are going to discuss today. So if nothing is clear or if anything is not clear, please stop me now. Okay, I will then proceed. Then came the idea of microarrays, and in hindsight I realized maybe I should not discuss microarrays because this is slightly going in a tangent, in the sense that it is solving one very specific purpose which we will cover when we cover the RNA sequencing part. So for today's class I'm going to skip, because we will cover it once we start talking about RNA-seq in 2 or 3 lectures down the line. But what then came or what happened then is second generation sequencing, and there is only one way to do it right, which is to watch this video. The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a bond, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and
emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is flushed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized, and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the read 2 sequencing primer. As with read 1, the sequencing steps are repeated until the desired read length is achieved. The read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variance identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in base-based sequence hub.
Okay. So this video, the first time you watch it, it'll be like it's a mouthful, right? It's a lot of steps going on. You'll not remember what is going on. Which music is cooler? Previous one, right? Previous one definitely has cooler music. We will cover the specifics of all what exactly we discuss in this video in this class. So it's okay if you did not get like full on each step, but hopefully you'll remember the picture. So just remember the picture, and now we're going to break it down and see what happens, right? So one reason I wanted to motivate this slide is actually what we are going to do carry forward from here is actually an image processing problem. We are doing biology and we are trying to sort of read the signal out of those sequences, but the signal that we are actually reading looks like an image, and that's where the algorithms come into play. How to go from these images to individual bases, that's where the innovation happens. So broadly, this technology or this family of technologies, it's called it has a proprietary name, sequencing by synthesis, and we'll see this why it's called so in just a bit. The central premise of why and how it works is first how it works. It starts with this genomic DNA. You shear it off, like you cut the DNA, you chop it off, you chop DNA into short fragments, and that's what the second step is. That's what the fragmented DNA stands for. Once you have this fragmented DNA, you will add some special sequences. These sequences are called adapters, which is, for example, highlighted here in purple. And the meaning of the adapters is very, very simple. There is a forward adapter and there is a reverse adapter. So we'll call it P5. It doesn't matter what we call, but there are two adapters that you have to keep into account. What is the goal, right? So the goal is that once we have this long sequence, we would want to sequence it. If we had a technology which we could read the entire thing at once, that would have been fantastic. However, that does not exist. So we try to make away with that problem by just cutting this down into short fragments. Once we do this short fragments, I want in some ways, like what is the problem now? There is only one copy of DNA that you have. Sequencing something which is only one, extremely hard, right? Like even if you make these copies, I will only have one molecule to detect. Extremely hard. You can just end up missing it by choice. What is the solution? Yeah, do some sort of PCR, right? To try to amplify it somehow. Now that's where the innovation comes. One sort of obsolete way, well, not obsolete, but like one way to do this would be you will take these short fragments and you'll run PCR. How long? You have, let's say, let's do some calculations. 3 to the power 10 to 3 to 10 to the power 9 bases. Let's say each of these fragments is 100 nucleotide long. So I have, how many fragments do I have? 10 to the power 7. How much time will it take to make 30 cycles, to make 32 copies of this? Anyone? Problem is clear. We have a DNA. I have only one copy. I'm going to make multiple copies of it. I know this. I know the technology, which is called PCR. I'm going to use it. What is the bottleneck there? So let's say we have a very fast PCR. Okay, that fast PCR gives us a turnaround time of 30 minutes. Okay, you have each of these as individual molecules. How much overall, how many PCR machines do you need? How many molecules can you do at once? I don't know the number, but it's going to be very large. I don't know how best to do it. But even if I brought a PCR here, right? So this is your PCR machine. It will give me a lot of these molecules, but it's going to be very cumbersome. Like, you know, me having to sort of somehow keep track of where, like having to come up with primers for each of them, very, very hard, right? Like I would have to come up with primers for each of these sequences, which is hard, like because, you know, like this is DNA, so DNA will change based on which organism you're working on. It's not as conserved or as same as protein sequences are. So this is not going to work out in the long run, right? Like even if you somehow make a very magical PCR for 5 minutes, this problem will remain. So how is this? So the technology, this problem, this sequencing technology tries to solve is how to get rid of this, because this is the bottleneck step. And the way this does this is extremely, extremely smart. And I have no idea how somebody would think about this, right? Like it's a work of 3 scientists primarily, and they won the breakthrough prize in 2022, which I'm going to highlight in just a bit. But how they thought about this, and you know, it's like one of those ideas which I still have no idea why and how somebody would think about this. Okay, so let's talk about it. Like what exactly does it do? So we cannot do this PCR outside, so we have to invent. And one way this mechanism invents is uses this technology or uses this very simple idea that I'm going to take a glass slide, and in this glass slide, I'm going to put spots. Okay, what are these spots? These spots are nothing but like these are special sort of sequences or special sort of just pores in your glass cell. And within those pores, I'm going to zoom it a bit, and we'll do all this in. And what it has is it has some very special sequences which are tethered to it, and these are called your These are called your these are complementary to your adapter sequences. So that when I take this sequence and I pass it through this flow cell, it will have a reverse complementary hybridization with what is tethered to my glass, or what's called the flow cell. This is the flow cell. So each pore, it's a pore. That pore has, your DNA can actually literally flow through it, right? So you take this emulsion, you flow your stuff through this flow cell, which is basically a flow cell, which is basically a glass slide which has pores inside, and within those pores you have these special sequences. Right, so think of this as you're flowing in this tunnel direction, and in those tunnels you have multiple sort of these projections out. These projections can reverse complement with your DNA, and once you do that, there is a very interesting stuff that is going to happen next. But each of this pore can have multiple such sequences. So multiple DNA strands can come in and just get tethered there. Okay, is that clear? What is happening? Okay, once this happens, what essentially the machine, or what essentially the way the chemistry is designed to do, is it uses again the ideas of DNTPs and DDNTPs, but in a very, very smart fashion. What we saw in the case of the Sanger sequencing was there was something called DDNTPs, which when incorporated will not allow extension. There are no, specifically there are no DDNTPs here. All of them are like sequencing by synthesis. So we are sequencing, we are going to synthesize the DNA. All of these are your natural DNTPs, but they have a fluorophore attached to it. So there is a special signal, again, red, green, blue, and yellow, which is attached to each of these bases, which is what you see in the last end, the last slide. At each point, you have a strand. Let me first talk about what is the principle on which it works, and then we'll see why we need to make it work like this. So imagine you had your sequence, which has got tethered here. You will add your polymerase, and as you add your polymerase, because these sequences are already known. Why? Because these are the primers. So this is called the P5, and this is called the P7. Just because there is some direction to it, but otherwise for this class it doesn't matter what exactly they are. So let's say here I incorporate an A. When an A gets incorporated, it will emit a signal. And that signal is being read by a very smart camera. There are cameras, like as you saw, it's an image processing problem. So what you will essentially see is in a block, you will see for each pore that you have, what kind of signals are being emitted. At each single time when a base is incorporated, the mechanism is so designed that only one base can be incorporated at a time. Okay, so the camera will see that, okay, a ping signal has been emitted, so it must be an A. So it has a fluorophore attached to it, which is a special kind of a signal which is emitting which is a special kind of attachment that you have done to your DNTP, which when incorporated will emit your red signal overall. And that's what you're focusing on, this particular pore at that particular time. Yeah, so you have your base, right? So the base looks like this kind of structure, right? It's a ring kind of a structure. You can add a molecule which has a signal. So this will emit, let's say, orange in this case. There are signals like this, which you can actually deposit on anything, which will emit a particular kind of a signal. Now at this point, the reaction will stop. The reaction will stop until a wash round will remove the fluorophore which was attached to it. So at each step, the polymerase is waiting for one wash cycle. So some reagents will be thrown away again along these pores. This will get removed, and now this base is ready for incorporating another thing. So it's like a DDNTP, but it is reusable. If I had incorporated a DDNTP, my reaction would have stopped. I have incorporated a normal base along with a fluorophore, which is washable. I can remove it. If I remove it, it allows me to get or to bind to another or extend in this particular direction. That is innovation number one. At this point, from each of these pores, right, like multiple fragments are being sequenced at one time. Yeah. How do you wash it? Yeah, so the purpose of what is the purpose of washing, right? So the reason for washing is, if as long as that particular molecule is there, it's still going to prevent its extension. So if you have a fluorophore attached molecule which gets incorporated, this extension is still blocked. It almost acts like a DDNTP, which doesn't allow extension. DDNTP, the change is permanent, so it will not allow any further incorporation. But in this case, I can do a wash and it will remove it. This gets removed, and now it's ready for another round of incorporation. Okay. So that's innovation number one. We will see this in as we go along as to what exactly happens in just a bit. However, if you want to detect this, again, if there was only one molecule, right? If there's only one molecule, detecting that signal is going to be super noisy. So you need multiple such molecules, and all of them need to be in sync. And that's where this idea of doing PCR on the plate itself comes. So while I showed you what happens in terms of the principle, which allows you to read each base at a time, if you only read one molecule, the signal to noise ratio will be extremely small, because it's, at the end of the day, it's like one small molecule, right? That's nothing there. So the way it does this is, at each spot, it's multiple molecules which are being read. It's not one molecule. Yeah. Yeah, fair enough. So at each point, how do you incorporate, how do you ensure that each molecule is only being read, the same molecule is being read in one particular fashion? We will see this in just a bit. I will answer that question. Okay. So in order to do this, what essentially happens is, there is this idea that if I make multiple copies of each fragment at each location, I should be able to tell on a general basis which bases are being incorporated. So if I had, rather than one molecule, if I had multiple molecules which look like this, and if all of them emitted a same signal, it would help me increase the number of reactions that I can do. So the way this is done is by doing what's called bridge amplification. What is bridge amplification? So the moment the molecule lands up here, let's say again this is another pore, it has a particular sequence which it tethered to, but you also had attached a second adapter sequence here. Now the way these adapter sequences are designed is, there is going to be another adapter sequence in its vicinity. So it's very smartly designed, which allows it to reverse complement, right? So this is your pink. It tethers to a sequence which is complementary to it. So we'll call it again pink. There is another end, which is the 3' end, which has yellow. And that yellow has, there is another pore, there's another sequence in the vicinity which has a yellow attached to it. This will allow it to bend over. The reason this happens is because molecules are looking for more stability, and any sort of reverse base pairing or complementarity will bring more stability. Because of this, this molecule that you started with is now going to bend over and create a bridge. The reason this is able to happen is because you have these sequences. These are your adapter sequences, which allow you to, which enable this overall. Now you will do another round of quenching. So now you have, in process, you have another molecule. Sorry, I should use the same color. The same molecule can now stand. Now I'll come to it in just a bit. But you have a reverse polymerase sitting here, which will make another copy of this sequence. And if you quench it, now you will end up with 2 copies of the DNA, and you can keep repeating this cycle. So what we were able to do outside in terms of our PCR, by making multiple copies of the molecule, we can do this on the channel here, on the reporter channel here that we have. Does that idea make sense? And we'll discuss some of the questions that Nakul asked. How does that happen? Does that idea make sense? Yeah. Yeah, we'll talk about how exactly the sequencing is taken care of. Maybe I'll go back to the video, because that does slightly more justice to than me showing you on the board. But the forward and the reverse, so let's talk about it. So the way sequencing works, right, when you get the data, like what does it look like? There are 2 ways that I could just only sequence one of this part of this DNA, which is called, what you get out of the end, what you will get as a computational person is what are called reads. These reads are nothing but 100, they're called read because, well, you're reading the DNA. They will literally look like this kind of stuff. and there will be multiple of this in each file. There will be millions to billions of such short fragments that you will get. This is, you can get it in one file, and that's called read one, which means that you're only reading each copy of the molecule at once, which means that you're only reading it in one particular direction. There is another version, which is the smarter version, which what the video was also about, which also does something more smartly. It says that while this is good, I can give you slightly more information, because I can not only sequence the forward part, but I'm also going to sequence the reverse part. Here, this sequence came from just doing a read through of one particular fragment of the DNA. Now imagine that this reaction, right? So we can still imagine this is what happened, and we have our, if this was to go in this direction, I will have my DNA which would look like this, sorry, yellow, and my other DNA is going to look like this. First time, you're going to sequence this and call this, I will use a white one. I will sequence this and I'm going to call it R1, right? So of course, sequencing happens, let's say, in this direction, right? So there's a specific direction in which the read through will happen, because the polymerase can only work in, which direction does polymerase work? So this is your DNA, right? You sequence always in the other direction, which is 5' for that particular sequence, and 3' in this direction. So this is the molecule that you're sequencing, but you will always sequence in the other direction. This is because that's how the polymerase works. So let's say for this molecule, the 5' is here and the 3' is here. What is the corresponding 5' and 3' for that sequence? Yeah, top to bottom. It's as simple as that. Okay. Now, if this is the direction, I will now fix this. I will do a read through in this direction, 5' to 3'. I will do sequencing in this direction. This becomes my R1. And similarly, in this case, I'm going to sequence in this direction, and that becomes my R2. That is called paired end sequencing. I have a choice what I want to do. I can tell it. I have a way, we'll see the video again, and I will tell you at what point you can stop the reaction. If you stop the reaction at this step, you only get one read, which is fine, which is great. But you can keep one more round. Of course, it's going to be slightly more expensive now, because you'll have to again add some more reagents and so on. But now you get two reads for the same fragment of the DNA. So this creates a lot of confusion, and for the first time, actually this is never usually taught in classes. This is another problem, and this is stuff that you always pick up on your own. But for the longest time, this confuses a lot of people. What is R1 and what is R2? So imagine this is the fragment of DNA that you're sequencing. What is R1 doing? So of course, this could have been, let's say, when you shear the DNA, you don't actually shear it to 100, right? You can shear it to 1000, you can shear it to 2000, whatever, it doesn't matter. How much you can sequence, that's usually 100 or 200 base pairs, because that is limited by the technology. And we will see this in just a bit, why we can only sequence a few of them. What is your R1 is this sequence. It's called paired end sequencing. Doesn't mean that this is the other end that you're sequencing. What you're sequencing, if that part was clear, this should not create any confusion, is you're sequencing the other end of this long fragment, right? So this is the fragment that you created. You're sequencing 100 nucleotides from here. You're sequencing 100 nucleotides from here. How much do I have in between? 300. And if this molecule did not capture those 300, there will be some other molecule which would capture it. Because when you shear it, right, you also have multiple possible ways of shearing it and so on. right? So it is not that that is going to get not there's never going to be sequenced. It will get sequenced. For that molecule, no. The entire length, that's a good question. We will come to it in just a bit. But the entire length, it's a condition of how much your sequencing can happen. If my sequencing length is going to be 100 nucleotides, and if this fragment was less than 100, I will sequence everything. But it's usually going to be longer. And that's why you're not going to be ever be able to sequence. That's a very important question, because I'm going to ask a quiz question based on that. We will see that question in just a bit. Okay. Of why it's important to understand. But so far, it's been very messy. We will clear this up because there are nicer diagrams there. But any conceptual questions here? Yeah. Mm-hmm. Hmm. This one. Yeah. Yes. Yes. That is correct. How do you get R2? Fair enough, right? So Devansh's question is that if PCR itself is not going to be very efficient, then what you're promising here that I will be able to create from this thing, this entire thing, will I be able to create it in full? That remains questionable. Is that what your question is? So that's a good question. My short answer is in these control settings, you can assume that if your fragment size, and there are some specifications for what should be your input fragment size for different technologies, like you choose how much to cut before loading it up. So it has been tested that if you load certain fragment sequences, it is able to handle it, right? And you will shortly get your R2 in that case. That's what the commercial technology sort of guarantees you. It is still doing PCR, it's still doing it in some fancy fashion. There's one important distinction though, that everything that you're doing here, the synthesis is happening right there and then, right? And that you're able to read the signal directly. So that is one important distinction, because it's not just being able to detect the molecule, but you're actually detecting the sequences rather than in the PCR. So the short answer is in these cases, you can assume that that usually happens. That's my short answer. I don't know a detailed answer to that question. In how many cases do we see a fall through? One of the other challenging parts about this particular technology is it's not able to, this is a commercial technology, so not a lot of it as to how much is the failure rate and how many times the PCR will fail, I don't know, because those are usually not reported, because your stocks will go down if you report all that, right? So, okay, any other questions? Yeah. Yeah. Mm-hmm. Ah, good question. Okay, there is one important step that I actually missed, and I think, well, so this sequence, that's a good question. So when you actually load the fragment, right, I only told you about the pink and the yellow stuff, which is your adapters. But what is actually also loaded in is what's called an index, which is called the I5. There is another, let's say, dash stuff here, and there is another dash stuff here, which is called the I7 and the I5 indexes. So there is a particular step which let's go back to the video and watch it again now with this context, which you spend actually sequencing, and you retain what index it came from. That is your identity of the molecule, which DNA came from where. So when you add the DNA, while these are like common sequences, P5 and P7 are like adapter sequences, your indexes will be a lot more. And that index is the identity of which molecule it came from. And that is also the short answer to his question, that how can I tell you that which molecule came from where? Because when that incorporation happens, this incorporation, this I5 and I7 will be like a lot of them. But these together tell you which molecule you came from. Okay. So when you do what's called base calling, you take into account what is your I5 and I7, so you know which molecule you're talking about. And that's how you keep into account Does that answer your question as well? I skipped over that detail deliberately because it becomes very technical for a first thing. But overall, what you're doing is you're taking the sequences, you're adding these adapter sequences, which are common, like, there is a fixed sequence that you're adding to all of them. But with I5 and I7, there is another special sequences that you're adding again to each shared molecule DNA, which gives that molecule its unique identity in some sense. And your molecule, when you are going to do this step, you also spend some time sequencing the I5 and the I7. That will tell you at the end, computationally, you can tell which data came from which molecule, because all of the R1 and R2 will have an I5 and an I7, which will correspond to one particular molecule. Yeah. Yeah, there are 2000. So I forget the length. The P5 and the P7 will be usually around 12 to 16. It depends on the technology. So Illumina has different flow channels and it changes with that, but I think it's 16 nucleotides. So that gives you a lot of permutation, right? But that is going to be fixed, so it doesn't matter. But with I5 and I7, you can go up to, so don't quote me on the numbers, but it is, I think, of the same order, 16 to 20 nucleotides long, which is a lot of molecules, right? So it's 4 to the power 20 that I'm talking about overall. So you have enough to sort of be able to detect everything that goes on. So you don't remove them. In some cases, when we start dealing with the data, you'll see that people also deposit these sequences. I5 and I7 are also sequenced in that fashion. No, it will not, because that is something which is taken care of before you get your R1 and R2. So your R1 and R2 are just DNA sequences. Ideally, they're not supposed to carry your I5 and I7 as such. Yeah. So, but you know the I5 and I7 indexes to begin with, right? Like these are special K-mer sequences which you already know what their sequence is. So when you're going to base call, you can actually remove them, and same for your adapter sequences, right? So your adapters also will get sequenced. I will talk about it in just a bit. How do you actually take into account how to remove them? Yeah. Yeah. Okay. From the I5, I7, we got a multiple. From starting, we got the four might have been there. Yeah. And now when as we proceed, someone will which some case will some cases will attack and will give some bit lights for us to detect. But again, that's random. Once the step starts, it will be hard to tell which of the fragment is in which part. Like, are they bridging or are they? But the I5, the P5 have fixed length. So you know how much to read and how much to discard, right? You know how much at each point, how much should you read, and because these are towards the end. So if I told you that 10 base pairs is adapter, 10 base pairs is I5, then you know that the first 20 are not actually your DNA. You know that beforehand. Right. Mm-hmm. But as it proceeds, yeah. We cannot control, okay, if it has which first or if it has complementary basis being attached with so it is very much in sync. So at each step, you know what is the reaction that is going on. The read of the camera will not happen unless it knows that it is at the synthesis step, which is that it is actually incorporating those bases, reading them in this direction. Because what you put in, and this is what the machine does actually, what you put in at each step is determined by the cycle, right? So if you're doing, of course, the extension, the bridge amplification stuff, you don't need fluorophores. So those bases will not be the fluorophore sequences. They will be your usual, good question. Those will be your DNTPs, normal DNTPs. So they will keep incorporated. I'm not going to emit any signal in this step. I will only emit signal when I am reading in this particular direction, and I also know at what step this has to begin. Okay? Yeah. So this is all, again, this is commercial. So it is very tightly controlled, right? Like this is, think of this as it knows at what step what needs to be injected into the flow cell. So it really is very, very smart in that fashion. How does that happen? What is the algorithm? That unfortunately I don't know. But to think of it very briefly, if you know how much time each cycle will take, you can actually determine at what time you should inject what. Okay. Yeah. Yeah, so you do this in step. Again, a good question. You do this in step. I'm now going to play the video again and all of your questions will be answered. You do this in step. You first do R1, you wash it off, and then you do R2. Right? So, okay. Any other questions? Good questions. I think this is how that's, so now replaying that video will be much more clearer. Okay. Any other questions? So what is the paired end? It's a misnomer in that sense, right? It's not the same sequence being read in two different directions. It's the same fragment which is being read in two different directions. So these reads are paired to this fragment. That's why paired end sequencing. Regardless, they are in different steps. Yes, regardless, they are different steps, but you're ensuring that this is the same molecule, right? Because of, again, you have these indexes of to where they came from where. Okay. Any other questions? Okay, so this is very briefly, right, like all the steps that when I play the video again will be a lot, but very broadly what is happening is you have these adapters. This is the simplified version, no indexes here. You have this flow cell. This flow cell already has this pink and the magenta adapters attached to it, so you can reverse complement. When this happens, at each point you can do bridge, each point there's a possibility of bridge amplification. It forms a bridge and then amplifies. This is your PCR. This is the innovation number 1, which allows you to make, or innovation number 2, whichever, I forgot which innovation was first, but this allows you to make multiple copies of the DNA. This is required because once you start to read the signal, you want it to be very, very sharp. And this is what gives you very, very sharp signal, because multiple molecules are detecting the same thing. So when you do the base calling, which basically is an algorithmic step, you saw that random image, right? In the beginning, that's what the algorithm sees. But it knows that if I have an I5 and an I7 index at each point, what molecule is emitting what, and hence I should assign it an A, a C, a T, or a G. That's what this allows you to do. That's why this particular step is required. And after that, you have a very precise camera, which is always reading all these signals towards when the incorporation happens. So adapters, like I described a lot of these words, but what are adapters? These are short pieces of double-stranded DNA with predetermined sequences. You know the sequences of your adapters. They're not surprise to you. You will be able to tell in a given data whether there are adapters present or not, because these sequences are already known, right? So they are first ligated to the double strand version, and then you're always dealing with single strand in sequencing world, right? Like double strands are complicated. You always sort of give it enough temperature so that they get denatured and you're only sequencing one strand. And of course, they can hybridize to the flow cell, right? So this is what it looks like. This is your fragment. This is your 5' adapter, 3' adapter, or P5 and P7, which I called. And that's what is going to get and sit on the flow cell. Why do we? Why do we need indexes? The indexes is to identify that this molecule is one, right? So the indexes is the identity of that molecule. No, the adapters are all the same. Every DNA fragment gets exactly the same adapter. There is no identity attached to it, right? So this will be like a, whatever, 12 to 16 base pair adapter, exactly same sequence to all the molecules. Okay, no innovation there. I5, I7, lot more permutations and combinations. That's why. Okay, so this is what happens in terms of the flow cells. This is a flow cell. You have oligonucleotides already tethered to it. Adapter will come. This is going to do a reverse complementary reaction, and that's what is going to sit there. This is your, of course, one of your step 1 when you're doing the immobilization. And then comes this innovation, which is going to do the bend over reaction, right? This is what is called bridge amplification. So this molecule that you took with the red and the blue adapter, the blue one is going to bend over. You will do the reverse transcription. So you will again make one more strand. You will do what's called cluster generation. So you do one more round of temperature heating, and these molecules which are forming these kind of bands will come together and stand straight on this spot. And if you keep doing this cycle multiple times, you will have a lot of molecules at once. So this is in place PCR, fast PCR reaction, basically, working with very, very small fragments. Okay. Any questions about this? Bridge amplification, bridge and amplifying. That's why bridge amplification. Okay, then comes the step of nucleotide addition. So how we are doing this? At first step, you immobilize the DNA because you have adapters. It tethers to your sequences, which are already there on your flow cells, right? So this is again to answer Ansh's question. These are all same actually. Like these are all same, and they reverse complementary, do reverse complementation to your adapter. Adapter sequence is known. I will tell you what the adapter sequence is. You can also find out in your sequencing data if you, it's a pattern matching problem. So all of them will have exactly the same sequence, right? So it's a, maybe we'll do it as an exercise in the assignment. Second step, doing bridge amplification, amplifying the molecules. And finally, now is the step that starts. This is innovation number 1 or 2. Two key innovations. One is bridge amplification. Second is sequencing by synthesis, which is what this particular step does. So all of this has happened, right? You have had, you have made enough molecules which you have duplicated. Now is the time to sequence them. To sequence them, you again add, you have adapters. So you know what, you know, your primer is already decided, because very simple, right? Like it's going to just be reverse complementary to the adapters that you had. You add it along with your ribosome, along with your polymerase and your bases, which has a fluorescent blocking group. This blocking group is very similar to what we saw in DDNTPs. Rather than an OH being replaced by an H, this is a fluorescent molecule sitting there, which prevents addition of any other base. So this is one base, this is A. Usually it will, it can have anything next to it. But if you have a fluorophore, it cannot extend in this direction. It will of course have something else sitting here. But if you have a fluorescent molecule sitting here, it will not extend. Instead, it will emit this blue light as it gets incorporated, which is what the smart camera, which is sitting on the top, is going to read. This camera is going to read one molecule at a time, right? So it doesn't matter where exactly these molecules are coming from. It's very, very precise, and you know their index. As you can figure out from their indexes which molecule corresponds to what. At each point, one single base will get incorporated because it's emitting a signal. Once that happens, you're going to wash the stuff so that the fluorescent molecules gets washed off. Once it gets washed off, now this is open for extension. So another base, let's say C or G or T, can come and extend in this direction because I have washed this fluorescent molecule. Is that clear? Any questions? Very fancy stuff, right? I have no idea how they thought about this. Okay, and this cycle keeps on repeating multiple times. You have 3 channels or 4 channels, what you can read your signal, A, C, T, and G, 4 channels. Yeah, so the 3' OH, which was removed by the fluorescent channel, is regenerated, and a new cycle begins with the addition of nucleotides, right? So the difference between with Sanger sequencing is, in Sanger sequencing, I would have to waste making different copies of the same molecule. Here, I'm using one same molecule to do one full read through. In Sanger sequencing, I would have made multiple such copies of different length and then have read through it. This is sequencing by synthesis. I'm synthesizing and I'm reading at the same time. That's what the innovation is. This is how broadly the class of technology which is now called Illumina, but was previously called Solexa, works. This is your Illumina sequencing. Okay. So at each step, very briefly, this is what is happening. You have one cycle, you see a green and a blue, so you can read that this is C or this is G, and you keep reading at each point until you end the molecule. And this is what your signal at each spot looks like. This is one of the reads, this is another of the reads. Not R1 or R2 at this point, doesn't matter, but these are different molecules which are being sequenced. Okay. Any questions? Any questions? Sorry. Okay, this is Shankar Balasubramaniam and David Kleinerman, along with Pascal Meyer, won the 2022 Breakthrough Prize. Any idea what is Breakthrough Prize? It's a big deal, actually. You might not have heard because it's very recent. So it's supposed to be the Nobel, in some sense, Nobel Prize for, of course, making breakthrough innovations. This was started by a very interesting collaboration between Sergey Brin, Priscilla Chan, Mark Zuckerberg, forgetting a couple of names, Annie Wojcicki, the founder of 23andMe. I think I'm pronouncing her last name wrong. And I'm missing one more person. He is from Israel. He's an entrepreneur. So they pool money and they run a Breakthrough Prize. And 2022 winners was these three people for their seminal contribution. Okay, while some of them are biochemists, you will realize that once you do the biochemistry right, it is a computational problem overall. Okay, so there are some challenges. One is the read length is 250 base pairs. And when there is one more step which requires some thinking, which is at each step you need to wait to remove that 3' OH or the 3' fluorophore group, and that reduces the processability of the polymerase. So it actually reduces how fast you can sequence, because at each step you're waiting till that fluorophore signal can be washed off, and then only you can extend, and that actually causes some delay. Because of this, the polymerase has to repeatedly pause. So what is the polymerase? So it's extending A, but now it cannot extend, so it has to wait here because I need to wash off that OH, and then it's going to increase. And because of this, it results in what are called shorter reads or shorter fragments. How long can I keep doing it, right? So I started from a 500 nucleotide sequence, which I wanted to sequence, but I cannot go there because at each step I am making multiple pauses, right? So I incorporated an A and I paused because I have a blue stuff attached to it. I incorporate to be able to incorporate the next step, I will have to remove this blue arm and then increase and then keep going. And that prevents it to actually do what's called long read sequencing. I cannot do a full-on DNA fragment, which is like 2000 base pairs at once, because it's an extremely very slow reaction, right? So that is one limitation. Now I'll come to Nakul's question or somebody's question, I forgot, which was about, whose question was it? Adapters. Yeah, your question, sorry. Yeah. So Apurva's question about what happens if you have adapters and if you want to, if you have a longer or a shorter fragment. So let me pose the question first. Let's watch the video and then you will help me answer that question. We'll just rewatch some part of the video. This class is always missing either the duster or the chalk. Okay, I will use the board. So, what we have is we have a very long molecule, or let's take two scenarios, right? So this is 500 nucleotide long molecule, and I have, of course, I have adapters, and I know there's no point sequencing it. There's no point sequencing it because I added them, so I know their sequences, right? So ideally I don't want to sequence them. And that's what the technology also tries to optimize for, is usually the sequence that you're going to get will start beyond 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 the adapter. Let me use a different word. So this is where sequencing begins. And usually the promise of this technology is, hey, I'm going to give you 100 nucleotides. These 100 nucleotides in your file would look like this. Each of them is going to be 100. This is called a FASTQ file. Fast. So we studied FASTA, which was nucleotide sequences. This is FASTQ. FASTQ because when it actually, one thing I omitted is when it tells you that this base is A, C, T, A, this could be whatever, X, Y, Z, something. It also tells me what is the quality associated with it. Is it 10? Is it 50? Is it 10? Is it 100? Which tells me, which is basically a way for it to tell me that I'm confident, 100% confident that this base is A. I'm only 10% confident this is A. I'm only 50% confident this is C. We will see this in just a bit, but that's why it's called the FASTQ file. All of this is a 100 nucleotide base pair sequence. So if it is 100, if it is 500 nucleotides, of course, it's all good, right? What will happen, however, if you had only, I will still have the adapter, but let's say I only have 75 nucleotide long sequence. What do you think will happen? The technology's promise is to give you, like this record cannot go below 100 nucleotides. That's the promise of the technology. Okay, one way is I'm going to say some random sequences. Think about it from first principles, right? So how is the read through happening? The read through, well, I should do some justice to this, but okay, it might not bend, but the polymerase is reading in this direction. Okay, what happens if I have a very small sequence? Okay, we'll come back to this question. Let's watch parts of the video again.
Amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, generated similar to the first read.
Okay, so that's your index generation step, right? So this is where you're actually at this point you're just generating the index sequence, the I5 and the I7 indexes. One important information which I sort of actually very quickly rushed over is there's one more round of washing that happens so that you only keep one strand of each fragment, which is a 5' to 3' or whatever, whichever strand you got from. Yes. So the first step of washing is you want to remove the fluorophore signal, right? So that washing will get rid of the fluorophore so that the next round of reaction can happen. Once it has reached towards the end, right, a full-on wash happens to remove the entire reed. That is your one version of your reed, and then you will index, you will you'll read the index, you will sequence the index of that particular molecule. So you know which molecule is coming from where, essentially. So it's very intricate process, but what you should remember, however, is that you have been able to tell which molecule you're sequencing. You have been able to tell what is the sequence of that molecule, and you've been able to tell it whether it's coming from, whether you're sequencing the forward part of it or the reverse part of it overall. So the reason you have to do the first wash is because you want to retain what's called the strand specificity, which will only become contextual when we talk about RNA sequencing. In DNA, it doesn't matter because whether you sequence the forward or the reverse, it's fine. But there are how an RNA is expressed, right? So if you have uh, yeah, I will talk about it. Just give me 1 second. So this is your 5' and 3'. Okay. Now, how a gene is expressed when the polymerase reads and expresses this gene, right? It could either read in this direction and produce it, or it could go to the opposite DNA strand and read in this direction and produce it. So if I want to sequence, right? So again, the 5' and 3' have switched here. This is 5' and 3' of this molecule, and this is 5' and this is 3' of this molecule. I want to retain the strand specificity, which means that I want to retain whether, let's say, this is called, let's say, the positive strand and this is called the negative strand. One way for me to do is, let me just sequence everything and it's all good. But I want to also retain if this came from a particular strand, if I did not do that first wash that they talked about, right? Let's go back to it.
Clustering is a process where each fragment on the surface. This oligo is complementary to the adapter region on one of the fragment strands. The molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule.
So if I wanted to, of course, I can retain all these two strands and still do my stuff, right? And it's valid because these are two copies of each other. In DNA, it's fine because it doesn't matter. Like I really want to figure out the entire DNA sequence, so it doesn't matter. But if I do this, if I do and I retain both of them in a setting like this, okay, so what's going to happen? So I have the 3' and the 5. We will discuss this in RNA sequencing, but just to give you a gist, right? So if I retain both the copies, I cannot tell you whether it came from this direction or it came from the anti-direction. And it has a meaning. It has a very specific meaning whether the gene is produced in this direction or is it produced in this direction. If I keep both the molecules, I lose the power of being able to detect the strand specificity, which strand my molecule came from. I would like to retain it, and that's one of the reasons you sort of get wash away that particular cycle. There is, of course, you have to take care of this in the protocol as well, but that's something, even if you didn't understand it, it's fine. Don't worry about it. Okay, where were we? Yeah.
The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on a flow cell. Index 2 is read in the same manner as index 1.
Okay, so that's where, so there is a explicit step required for reading the indexes, which is going to tell you which molecule, what is the identity of that molecule that came from. It's not as reading the adapters, because adapter sequences are fixed, index sequences are not, right? So you did one round of read up in index 1 and second round of read up in index 2 in the other two different directions. That is your identity of the molecule.
Forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the read 2 sequencing primer.
So at this point, you sequenced read 1, which is that you did this read through. Now you're going to generate. I could have stopped the reaction here itself. I have a choice. I don't want to waste my reagents. I don't feel the need to sequence the second strand or the other strand, because the molecule, there is some redundancy in terms of the information that I'm capturing, right? Because it's the copy of the same molecule anyway. So you have a choice whether you want to generate R2 or read 2 or not. In this case, they show that you do generate R2. Okay? Any questions?
As with read 1, the sequencing steps are repeated until the desired read length is achieved. The read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices.
Okay, hopefully that was clear. Any questions? What happens to the mid, right? So you have sheared the DNA randomly, and overall, when you have sheared this particular DNA, there will be some other molecule that is going to capture that middle part overall, because while you have one copy of the DNA, there will be multiple. When you do the bridge amplification, that part itself will get, at some point, you will have molecules which are these cuts themselves. There's one more, actually, so I cheated a bit. There's one more step of PCR before you load on the samples. So you first make slightly more copies, then you randomly shear them. So it's not like it's a continuous, it's a discrete set of copies that you're making. So you're doing one round of PCR or few rounds of PCR, like 8 cycles or so on. You will generate random such stuff. So the hope is that at some point, all of the, you will have. But there are, like, you optimize your reagents and protocols and cycles in such a way that this is guaranteed that you will have enough coverage. So we'll cover coverage, and that time we will see how much does it get captured. But there are ways to determine how much coverage you need and how many rounds of amplification you need. Any other questions? Yeah. Yeah. No, you just have multiple things, right? So that's where the algorithmic part will come into play. So I've generated multiple such sheared versions. I will switch on this slide. So there are multiple such versions of things that you have generated, right? And now you're going to sequence. Some of them will get jackpot, and you'll sequence them. Once you sequence them, that's when the problem of figuring out which came from where begins. How should I put it so that where does it align? Which we will discuss in the next class, but that's exactly what happens. Okay, where did it come from? That's, again, where did it come from, right? So that's what the question is. Say that again. Yeah, you are taking all of them, right? Like, so the hope is of this shearing is that shearing part itself should ideally contain, like, I drew discrete versions of it, right? But you will shear it multiple times in some ways, basically, so that you will generate, let me draw it here. You will generate multiple such copies. So let's say this is your DNA, right? I will share it, and this is one way to generate the fragments. There is another way to generate the fragments, which is like this. There's another way. I will put all of them in through the sequencing part. Okay, and we will cover algorithmically how do we solve this problem. That okay, now I have this read. Where did it come from? Did it come from here, or did it come from here, and so on? That's a pattern matching problem. That's a algorithmic problem. We'll cover this in the next class. Yeah. Yes. So we will do that in the next class. We will do how to think about it, right? So at this point, all you get is like these. You don't know anything. Your sequencing only tells you this is a 100 base pair sequence. You figure out where it came from. I don't care. That's your job, and that's where we use algorithms to do it. Is that what your question was? Yes. So that will do in algorithm, right? We have lost the identity where it came from. I drew it as a discrete thing, but it's not discrete because it can come from anywhere actually, right? So it's a needle in the haystack, literally a needle in the haystack. Okay. Hmm. Yeah, so it's the complement, right? So all of this you have to keep into account, right? Like whatever you're getting, uh, the directionality is in some way, you're not never changing the directionality, but there will always be flip happening because 5 prime to 3 prime is the sequence and the read through always happens in the other direction. Yes. So that's called the first strand synthesis. Yes, you're only sequencing the first strand, right? And your algorithm again, right? As long as since you're not switching the directionality at any point, right? You're retaining only things which came from one particular direction. Well, you're retaining the original direction of the molecule. At no point the switch is happening. Your algorithm can handle where it came from. That's at the algorithmic step. It doesn't care about after that. Okay. Yes, it is. It is. It is. And you will tell me in the next class why you want to generate R2. Why waste resources? Any ideas? Any ideas? Why waste resources generating R2? Okay, one is RNA sequencing, but why? Why should I care? I want to save money. I don't want to generate. Okay, but how would R2 help you? How would R2 help you? R2 is just a sequence coming from the opposite direction, right? So, just to give you context again of R1 and R2, R1 molecule same, direction opposite. Why should I care? For cases where you are not able to.
You can get from R1 and R2 instead of just doing R1 and like small fragments of the molecule. When you're doing R1 and R2, you're probably going to get both sides of the molecule. So you have to do that. So you can stop your.
Yeah, it's covered in that step, right? So you're getting enough from both directions in some sense. But why waste resources to it? Okay. Hmm. Hmm. Okay. Anyway, yeah, Apurva. Sorry, remind me your name again. Aman. Yes. Yeah. Yes, but even if it is ANSI sensor sense, you're still doing it, right? You're still getting R1 or R2, right? You're still doing it. It is agnostic of R1 or antisense, sense and antisense. So what Apurva is saying is that sense and antisense, which is what I talked about, is that you want to retain this identity in some sense, but R1 and R2 does not really help you with that, right? Because you can do an R1 R2 for this molecule also and for this molecule also. Any other ideas? Think about what happens. So think about what happens once you get this data. What will you do with this data? Oh yeah, okay, yeah.
So if we, let's say we are doing it for R1 and we have two overlapping strands, and then we try to pattern match it. Then the extra data or the sequence we're getting is very little, as opposed to when you have R1 and R2, and let's say like this is pattern match, right? Once you reverse R2, so like you get more extra data.
Okay, so very good. So that's very very close, right? So let's say you have you have these sequences, huh? Right? Like you know that this is. Let me make it. Okay, let's say these are some special sequences somewhere embedded in your DNA. Now I got my data, so I start mapping R1. Now what I see is R1 maps here, like it is a match here, and it's a match here also. So algorithmically, it can map to two different locations. This is my genome, this is my R1, which is 100 nucleotide sequences. It can map to two different locations when I do the pattern matching, because it can happen, right? Like the chances of this happening is very small, but it can still happen. Here is where having an extra set sort of helping hands will be useful. If I knew that what is the corresponding R2 to it, I will go and try to map it as well. And when I try to map it, what I see is it maps also. But there is no sort of corresponding match close to this, right? Let's say the distance between them is, let's say, maybe 500, and the any other R2 match that I find is way beyond this. Let's say the distance between them is like 10 kilobytes. So it's extremely unlikely that R1 maps here and R2 maps somewhere very far away from this sequence, but I know that it likely originated from this fragment of DNA. So again, this is where your biology and usefulness of knowing this protocol will be useful, because you know that your most of the fragments have to be close, because we cannot go in opposite directions if the fragment is very very long. So R1 will map here, R2 is mapping here. I'm going to use this algorithmically. So having paired end data helps you map sequences much more confidently. That is what the idea of. That's why we waste money doing the paired end sequencing. Okay, that clear? Okay, I think we are. How much time? 2 minutes we have, right? 2 minutes. Okay. Okay, I guess I've already covered this, right? So this is called the insert size, the amount of stuff that you want to sequence. This will be longer. Your reads often will not be able to capture it. We will discuss this problem at some point, or think about this problem. I don't want to discuss the answer. What happens if it is shorter? And we'll quickly show you, this is called sequencing 3.0, the title of this lecture. So I cannot do justice without actually showing you what is the sequence 2.0. So if you give me 2 minutes, a video is more than the words. So I'm just going to play the video. And of course, when we meet next time, I will cover this. There's literally just one more slide that I have after this. Can I take 5 minutes?
Introducing the PacBio Sequel 2 system, powered by single molecule, real-time, or smart sequencing technology. Here's how smart sequencing works. First, from any sample type, ranging from viruses to vertebrates, DNA or RNA is isolated. Next, a SmartBell library is created by ligating adapters to double-stranded DNA, creating a circular template. Primer and polymerase are added to the library and is placed on the instrument for sequencing. At the core of smart sequencing is the SMART cell, which contains millions of tiny wells called zero-mode waveguides, or ZMWs. A single molecule of DNA is immobilized in the ZMWs, and as the polymerase incorporates labeled nucleotides, light is emitted. With this approach, nucleotide incorporation is measured in real time. With the Sequel 2 system, you can optimize your results with two sequencing modes. Use circular consensus sequencing mode to produce highly accurate long reads, known as hi-fi reads.
Okay, so what happened? Was it clear? What happened? So we were limited by short fragments, right? Like I'm getting only 100, 200. I don't, it's very hard. It makes my algorithms suffer because matching a 100 nucleotide sequence to 3 billion base pairs is hard problem. I would really want technology which can go as long as I want, right? So what I was sequencing was this much, but now I want this much. And that this much comes from one of the technologies that enables this is called pack bio, where there is no shearing required. You take the double strand of DNA, you ligate certain adapters to it, and then you make them sit through in what's called a zero mode waveguide. These are very small sort of wells which you saw. So again, you are using flow cells in some sense, but each flow cell is a well. That well is where you have your polymerase, the read through is happening, and it's emitting a molecule which is again a fluorophore signal at each step, and that is what the system is going to read through. Because of this ability that you're actually not shearing, you can go very, very long. You can go up to 8 to 10 kilobytes, and in fact now even up to 1 megabases with this technology. Now with this in mind, just give me 2 more minutes to actually, for people who missed there's no better way to end than at the music that's going to play now. So I don't need to explain this because it's going to be very, very simple, but we will start the next class just explaining this technology. Any questions about the previous technology? That's called pack bio. Sequencing, third generation of sequencing. The advantage being long molecules, the disadvantage being higher error rates. Why higher error rates? We'll cover in the next class, but please enjoy this video. Okay, so that was the nanopore technology. It's a sequencer that can sit on your palm, gives you 8 kilobytes, 8 kb to, I think now up to almost a half an MB worth of sequences. Very powerful. We will cover some of its applications in the next class, and then we actually begin. Right, so I know we are, this class is a hodgepodge in some sense, right, but we are now getting more into the computer science sort of thing. So once we start beginning the next class, that will focus on how, once you get this data, how to make sense out of it. Okay, and then we'll do a hands-on, and we'll cover RNA in the future lectures before the midterms. Thank you.